Hi, I'm Jane Larkindale, and I was asked to speak here about some of the work we've been doing through the Duchenne Regulatory Science Consortium, where we've been really working at trying to understand how to build optimal clinical trials. So the idea is to make clinical trials as short as possible, involve as few people as possible, and really still get to the point where at the end of the trial, we know whether a drug works or not. So we want to learn as much as we can from the trial while still keeping everything as efficient, quick, and effective as possible. So these are a bunch of graphs which probably look a little scary for those of you who don't like maths and numbers, but what they really show is that whatever endpoint we collect in clinical trials is extremely variable in the Duchenne population. And I think most of us who have hung out with people with Duchenne know how variable they are. They gain strength in early disease, they lose strength later on. But how much strength they gain and how much function they gain before they lose is variable from patient to patient. Some decline more quickly than others. So if you look at just a random selection of patients, their progression may look quite different to each other. And that's really shown here for each of these 6 outcome measures, where each little circle is a single observation. And basically, it looks like a great big mess of dots with no discernible pattern. The blue lines going through them show that there is a pattern under there. It's just so extremely variable from patient to patient. It's very hard to see in the greater population. And this is a problem when you're thinking about designing a clinical trial, because you either need to have an awful lot of patients or a very long time to see a significant change between patients on drug and patients not on drug. Even that's assuming the drug works. So this was the problem that we set out to try and resolve. Fortunately, despite that great big mess of variance in the population as a whole, if you look at each individual patient, then that's what's represented by each of these figures here, you can see the pattern is somewhat similar. Most patients in their early stages, under the age of 7, there's some increase in strength and some increase in ability to complete functional measures. And then this decreases over time. And while the peak, the age at which the peak occurs is different from patient to patient, and the rate of decline is different from patient to patient, the pattern is there. Furthermore, there's a very standard order of loss of function. So the age at which certain functions are lost is predictable relative to the age other functions are lost. And this all helps us put the information together to really understand how patients will progress in the absence of a therapy, and therefore will help us understand if a therapy is slowing that progression, stopping that progression, or improving a patient's function. And that was what we wanted to tease out and really understand in the Duchenne population is how can we determine what a group of patients would look like if they weren't on drug. So if they were on drug and the drug was having an effect, we could see that it was different, preferably using as few patients as possible and looking at them for as short a time as possible, while coming to a result that we could all agree upon. So traditionally, when people started running clinical trials in Duchenne and there wasn't much data out there, when somebody came to design a trial, they really were just looking into a crystal ball, or really their neighborhood clinical expert, who, based on their own experience, would say, well, I think the trial should be in this group of patients, it should run for this long, and if we look at this particular outcome measure, this will tell us whether the drug works, which sometimes worked, but quite often, some of the assumptions were wrong. It was based on a relatively small number of patients and observations. And we ended up with trials where at the end of the trial, none of us were quite sure whether a therapy was effective or not. What we want to do is to move into this new world that when you gaze into that crystal ball, it's full of all the information, everything we've learned from past trials, past studies, information from every patient in the population. What we really want to do is understand the population that's not being treated as well as we possibly can, so that when we add a treatment, we know if we've changed that progression of disease. Are we having a positive effect on the patients in that trial? And that is what we set out to do with the Duchenne Regulatory Science Consortium. So we're a nonprofit consortium. We consist of drug companies, academics, clinicians, Parent Project Muscular Dystrophy, and other nonprofit groups, a patient representative, and also representatives for the NIH and FDA, who all came together to decide how we could resolve this problem and how we can develop tools that are not just intuitively acceptable or acceptable on an academic basis, but could actually be accepted by the regulators and be used to approve drugs for Duchenne muscular dystrophy. So that was what we set out to do. we decided the way to address this issue was really to build complicated mathematical models of disease progression so we could understand how the disease changes as measured by different endpoints, understanding the population-wide variability in disease and the causes of that variability. We wanted to be able to understand subpopulations of patients. So particularly in early stage trials, you could choose a fairly tight group of patients, understand if the drug worked, and very quickly move into the longer, more sophisticated trials that are required for approval. And at the same time, we felt we needed to understand what clinical trials look like in this population. How many patients drop out of a trial on average? What's the placebo effect? How does that interact with the drug effect we hope to see? So really, when we get the results from a trial, we can fully understand whether that therapy works or not. So we're building a tool, which we call a clinical trial simulation tool, which really lets us run pretend trials on a computer first and say, if we have used these patients and we run it for this long and we have this number of patients in it, are we going to see the drug effect we see as a statistically significant change? If the answer is yes, great, we'll go ahead and run the trial. If the answer is no, we'll tweak those factors. Maybe it needs to run a little bit longer. Maybe we need a few more patients. Maybe we need a slightly different design, because by the time we come to actually run a trial in actual patients, we want to be sure that if the drug works the way we think it works, we're going to get a definitive answer. So how do we get definitive answers? We want to make sure we choose the right patients based on measurable parameters matched with the right endpoint. Now, the right endpoint for Joe might not be the same as the right endpoint for Tom, because they might be in different stages of disease. But there is probably an endpoint that we know about that's appropriate for measuring progression in each of those patients. So we need to make sure we have the right patients with the right endpoint. We follow them for long enough, power the trial appropriately. That means have enough patients in there, and understand what the placebo arms are likely to do. So hopefully we can reduce the number of patients on placebo and get, again, to an answer faster and using fewer patients. So this is very much a picture that I drew just to really try and explain this in a different way. So in a traditional trial, we're using all kinds of patients. We're not selecting particularly precisely who goes into the trial. We add a drug, and you can see in this untreated population, those are the blue dots in the top graph. You then add the drug and you get the purple dots. And I think most of us can see that that's moved the curve to the right compared to the average of the blue dots. But you can't really see it because there's so much variance in the population. All of those dots are overlapping. Instead, in a model-informed world, we understand which of the patients progress more quickly, which ones progress more slowly, those would be the green dots, and which ones are in the middle, those are the blue dots. And for the case of this experiment, we're looking only in that population of blue dots. Then we add the drug, and you can see those purple dots are very clearly separated from the blue dots. So the drug effect is the same, but by understanding that underlying variance in the population, we've gone from a trial that was not statistically significant and didn't really tell us if a drug worked to one that did. And this is the goal of what we're trying to do. Understand the variance of the population so we can choose the right patients, the right trial design, and the right endpoints to know as quickly as possible if a drug works. So this is actually language from our regulatory submission to both EMA and FDA of what are the use cases of these models that we're building. It's primarily to optimize phase 2 trial designs. If you remember, in drug development, we have phase 1 trials, which are safety studies, phase 2 studies, which really start getting us down the road and knowing if there's any efficacy, and phase 3 trials, which are bigger and really prove efficacy. The goal here is to get through that phase 2 as quickly and effectively as possible. Get an answer, is it worth going on to the longer, harder phase 3 trial as quickly as possible? Of course, the models can also be used to inform phase 3 trial design and hopefully reduce the size and length of those trials as well. They can also be used to optimize the control arms, choose appropriate control arms to be used in the trial, and to optimize potential novel trial design. So we maybe can look at different endpoints in different patients. We might be able to augment a placebo arm so we can reduce the amount of patients on placebo, and many, many other uses, because the more we understand about how our patients change over time, the better equipped we are to understand if a drug is changing that. So how are we doing this? Well, we're building mathematical models, so we love data. Data is hugely important. The more data we have on the bigger diversity of patients we have, the more we understand Duchenne as a whole, and the better we can design these trials. So de-risks, one of the first things we did was try to access as much data as we could from the community. And we now actually have 20 data sets in-house. They're not all on this slide. But we brought all of this data in. Some of them are from clinical trials, some of them are from natural histories, some of them are from clinical collections. And we standardized that data so we could integrate it all together into one place. And we can use that data then to build the models. We are very lucky in that many of our data donors or data contributors allow us to share some of that data, which means that data can also be used to answer other questions drug companies may have, research community may have, and to learn more about Duchenne. And something we're very, very passionate about is that data should be shared so we can learn as much as we can about our population and move research forwards as much as possible. And that's what the blue versus the white is on this table, is we can share those data sets that are in white. So in total, in our analysis data set to build our models, we have 1,137 patients. That's a lot of patients in a disease as rare as Duchenne. It's also 23,305 observations, because each of those patients have many more than one observation. And that makes it a big data set. Interestingly, we pulled our analysis data set about a year ago, and we have much more data than that now, and we'll be using the additional data as to validate the models and prove that they're generalizable across a broad population. So what do we do with this data? We take it and we model it. We build these mathematical models, and we understand how the endpoints we've chosen change based on a whole range of factors. What age is the patient? Is he on steroids? What mutation does he have? Does height and weight affect things? How about their earlier function? And this helps us predict what how these endpoints are going to change over time. And at the other end, we come out understanding the rate of disease worsening. We understand the trajectory, the rate, and the predictors of that. And it allows us to build this web clinical trial simulator. As my colleague likes to say, what we're doing is taking data and turning it into knowledge, something we can use to design better trials. I'm afraid I'm going to get a little mathematical here. And show you one of our 6 models, because we've looked at 6 different endpoints. And this is forced vital capacity, which of course is a respiratory measure, which changes relatively late in disease. You can see on the left-hand side what's called the training data set. This is the data we use to build the model. And you can see forced vital capacity increases and then it decreases, like many endpoints in Duchenne. We then took that model and put it into some of the data we've reserved, called the test data set, to see if it predicted what we expected it to do in the test data set. And you can see that those curves are similar, but not identical. But what's really nice is when you go down to the right-hand side, you look at these things called goodness of fit plots, which compare, on the left-hand side, the population predicted forced vital capacity compared to what was actually observed. And you can see our predictions are pretty good. And then if you look on the far right, if you look at an individual patient, our predictions are very tight indeed. So these models really do a pretty good job of describing what we expect to happen to patients over time, which is really important in trying to understand trial design. So this is, again, our forced vital capacity models, which have now being turned into what we call a clinical trial simulation tool. And this is a much more user-friendly way of looking at these models for those of us who aren't statisticians and modelers. We can go in, choose our trial design, how many subjects, how long we want to follow them for, how frequently we're going to look at them. We enter the characteristics of that population they're going to our trial, what's their baseline forced vital capacity, what age range, various other parameters like which mutations they have if that affects the rate of change of forced vital capacity. We tell it what we think our drug is going to do, and that's based on previous experiments. And then we look at that, tell it to run a certain number of simulations, and it will show us what will happen in that trial. If it shows a significant difference between drug and placebo, great, we can move on. We can pull out what our patient population looked like. We can export the plots. We can look at the power estimates. We can export those graphs, which tell us what that trial is supposed to look like. If the parameters don't work, we go back and we jiggle those bars until we have a trial that will work. This is what we're aiming at. We're not 100% there yet, but we're getting pretty close. So where are we? We have models. We have really pretty good models of each individual endpoint. The plans for that analysis has been reviewed by both FDA and EMA, who have told us to go ahead and build the models, that they generally agree with the plans. That's really important, because if the regulators won't accept these models, we can't use them a priori to design clinical trials, and it gives them the stamp of approval that these are something that can be used in drug development. We're currently finalizing our clinical trial simulation tools. There's still a few bugs in them, but we're getting close. And by the end of the year, we hope to have our final regulatory review with both FDA and EMA, at which point both the underlying models and the simulation tools will be made available to the community for use. Because, of course, we are a nonprofit organization, and our goal is and always has been to really help design better clinical trials so we can get answers about whether drugs work as quickly and efficiently as possible, so that we can get therapies to the patient population as quickly and efficiently as possible. We want to make the trials faster. We want to make the regulatory approval faster. We want to make the reimbursement faster. And by really understanding how disease changes in the population, how individual groups of patients are going to change, which endpoints are appropriate, and how many patients we need to look at to see a drug effect that we predict, will really get us there faster, because we want efficient, effective drugs for Duchenne muscular dystrophy. I would be remiss in ending this talk without thanking all of our data contributors, because, of course, we do not gather new data ourselves. We work with data for the community. And many, many groups, as I say, we now have 20 data sets, and many, many, many people have contributed to this analysis, particularly our modeling team and all of our consortium members. So a huge thank you to the whole consortium and to all of our data donors who have made this work possible. And if you have any questions about our work, feel free to send me an email at this address. I'm always happy to talk to people and help you understand what it is we do, why we're doing it, and how you can help us move this kind of work forwards. Thank you.